The small intestine is the main site for both digestion and absorption. Absorption includes absorption of nutrients as well as absorption of water. And we will see that the majority of nutrients and water, including the large amounts of water that are used in the secretions of the digestive system, will be reabsorbed here. The small intestine is named because it is smaller in diameter than the large intestine, but it is longer. The small intestine is approximately 1 inch in diameter compared to the large intestine, which is about 2.5 to 2.75 of an inch in diameter. The small intestine is much longer than the large intestine, and in a healthy individual, the small intestine is approximately 13 feet in length. The small intestine has 3 regions: the duodenum, the jejunum, and the ileum. The duodenum is the initial segment of the small intestine and is approximately 8 to 10 inches in length. It begins distally to the pyloric sphincter and curves around the pancreas. It is shown in the purple region in this diagram. The jejunum in blue is approximately 3 to 4 feet in length and is situated between the duodenum and the ileum, while the ileum in pink is the longest region of the small intestine, measuring about 6 to 7 feet in length. It is also the last region of the small intestine. It connects distally to the cecum of the large intestine, and a small valve called the ileocecal valve is situated between the ileum and the cecum. The small intestine has a massive surface area. This large surface area is due to 2 structural modifications. The first are the circular folds, which forces chyme to spiral through the lumen of the small intestine rather than taking a straight, direct path. The second are the finger-like projections of the mucosa that are called the villi. The villi contain capillaries for nutrient absorption, as well as lymphatic capillaries called lacteals, which carry absorbed lipids. The surfaces of the villi are not smooth, but are coated with microscopic projections that are called microvilli. Microvilli are small cytoplasmic extensions of the mucosal cells. The function of the microvilli is to help further increase the surface area of the mucosa of the small intestine, so that way absorption can be more efficient. Microvilli are also referred to as the brush border. So there are brush border enzymes that are bound to the surface of the microvilli that function in finalizing protein and carbohydrate digestion. We also saw one enzyme called enteropeptidase, which is a brush border enzyme that functions to activate trypsin from the inactive form called trypsinogen. The mucosa of the small intestine has many tiny pits lining it, and these pits are called intestinal crypts. The cells lining the crypt produce intestinal juice, which is a mixture of water and mucus that functions to carry the chyme. These intestinal crypts do resemble the gastric pits that we saw lining the stomach, but remember that the stomach does not function much in absorption, so the stomach does not have villi or microvilli within it. What cells are found in the mucosa of the small intestine? The most common cell type is the enterocyte. The enterocyte is a simple columnar epithelium which functions in absorption of both water and nutrients. Nearby enterocytes are joined to one another by tight junctions so that the enzymes in the digestive juices don't seep down into the deeper tissue layers and cause damage. In particular, protein digesting enzymes that are secreted by the pancreas and the small intestine are capable of digesting the muscle and connective tissue that's found in the walls of the small intestine. There are several types of cells that are located within the intestinal crypts. The goblet cells produce mucus. The enteroendocrine cells produce cholecystokinin or CCK and secretin. We see that an increase in the release of both CCK and secretin increases the amount of pancreatic juice that is secreted from the pancreas, as well as the amount of bile that's released from the gallbladder. The Paneth cells produce agents that kill microorganisms within the small intestine. In particular, they kill bacteria. And there are also populations of stem cells, that is, relatively unspecialized cells that undergo mitosis. Some of these stem cells will become more specialized cell types, such as the ones we just discussed. The intestinal crypts function to produce intestinal juice, which is a mixture of water, some mucus, and bicarbonate, which gives it a slightly alkaline pH. This alkaline pH is helpful for neutralizing the stomach acids that are mixed with the chyme. We see that approximately 2,000 milliliters of intestinal juice is produced daily, and we see that the presence of acidic chyme from the stomach stimulates the production of this intestinal juice. Now let's examine the digestive processes that occur in the small intestine. Both enzymatic digestion and absorption of nutrients are the two major digestive processes that are occurring within the small intestine. Recall that chyme from the stomach contains partially digested proteins as well as partially digested carbohydrates. This is because protein digestion began in the stomach and carbohydrate digestion began in the mouth, but neither of these digestion processes are completed by the time the chyme reaches the initial section, that is the duodenum of the small intestine. Chyme also contains undigested lipids or fats. This is because the small intestine is the place where bile is secreted, and bile helps to mechanically break up the lipids so that digestion by enzyme can occur much more easily. We see that absorption of nutrients is also a major function of the small intestine, and absorption typically takes between 3 to 6 hours once foods are completely digested by enzymes. So where do all of the digestive enzymes in the small intestine come from? We see that the liver is the source of bile, and I hesitate to put bile in this slide because recall that bile is not an enzyme. Rather, it acts like a detergent. It functions to emulsify lipids and fats, which is a type of mechanical breakdown that helps break up the lipid droplets into smaller pieces, so that way enzymes can digest them much more easily. The pancreas is a major source of enzymes such as amylase, trypsin, and lipases that are secreted into the small intestine, along with bicarbonate, which is not an enzyme but still functions to neutralize stomach acids. The enterocytes that are within the small intestine are also an important source of enzymes. We see that on their brush border, that is on the microvilli of the enterocytes, enzymes that are located there will finish carbohydrate and protein digestion that was started by the enzymes in the mouth and in the stomach. So recall that carbohydrate digestion begins in the mouth, thanks to the action of salivary amylase that's produced by the salivary glands. So once the enzyme salivary amylase has entered the stomach, along with the food that it was digesting, the amylase will be destroyed by stomach acids. So carbohydrate digestion will cease temporarily while food is in the stomach. Once the chyme enters the small intestine, pancreatic amylase will resume breaking down those starches. We also see that brush border enzymes will break down disaccharides, that is, substances like lactose, maltose, and sucrose, into their component monosaccharides. We see that lactose is digested into galactose and glucose. Maltose is digested into two glucose molecules, while sucrose is digested into glucose and fructose. We see that glucose and galactose are co-transported across the intestinal mucosa and into the bloodstream along with sodium. See that protein digestion will begin in the stomach with the enzyme pepsin, which is secreted by the gastric glands, and digestion of proteins will be completed in the small intestine thanks to the action of enzymes like trypsin from the pancreas and brush border enzymes. Generally, amino acids are absorbed with co-transport mechanisms along with sodium, just in the same way that we saw the carbohydrates or the monosaccharides being absorbed and co-transported with sodium. Finally, lipid digestion primarily occurs within the small intestine. We see that the salivary glands and the stomach do secrete some lipases, which are the enzymes that are digesting lipids, but these do very little to break down lipids in these structures, and the majority of lipid digestion occurs in the small intestine. We see that emulsification by bile is necessary in order to break down the lipids into smaller droplets, and then lipases from the pancreas help to finalize lipid digestion into fatty acids. The fatty acids can then cross the mucosa of the small intestine by diffusion because they can diffuse directly through the plasma membrane. Once they are inside of the enterocytes, they will be recombined into triglycerides, and those triglycerides will enter the lacteals. So it is important to note that chyme entry into the small intestine is very highly regulated. Only small volumes of chyme are permitted to enter. This is because chyme is hypertonic, that is, it has a tendency to draw water out of nearby tissues and into the lumen of the small intestine. Therefore, entry of chyme into the small intestine must be slow to prevent massive water loss by osmosis. The pH of the chyme is also very low, that is, it tends to be very acidic, and it must be increased or made more alkaline. We see that segmentation will typically move food through the small intestine after a meal while the food is being digested, because the movement of segmentation ensures that thorough digestion and absorption will occur. This allows the chyme to spend adequate amounts of time in the small intestine in order to be absorbed properly. Between meals, that is, after most nutrients have been absorbed from the chyme, peristalsis will move the indigestible food residues towards the large intestine.